Hi everyone, welcome to my channel Pure Biology. And before going to the topic, I say sorry for not giving any video in the previous week because election was occurring in my areas, so that's why I didn't give that. Okay. So today's topic is that sporophytic incompatibility. So generally incompatibility that are incompatibility is of two kinds. One is the gametophytic incompatibility and second one is the sporophytic incompatibility. So gametophytic incompatibility I have already explained in my channel, so you just check out. And this is the sporophytic incompatibility. Okay, today I have to explain to you. Okay, so the sporophytic incompatibility, that is the SSI, that occurs when one of two S alleles of the pollen producing sporophyte that matches one of the S allele of the stigma. So generally S allele present in both pollen and the stigma, and if one of two S alleles of the pollen that producing the sporophyte and matches one of the S allele of the stigma. So here this sporophytic incompatibility happens when one of the S allele that present in the pollen matches with the S allele that present on the stigma. Obviously S allele is not matches. Generally S allele produce the sporophyte that matches with the S alleles produce the sporophyte in the stigma. So sporophyte is the important thing, not the allele. Here the S allele that is important, and the S allele that present in the pollen that produce the sporophyte when it matches with the sporophyte produced by the stigma S allele. So here it is very important thing that one of two, that means if two alleles or two sporophytes are there and one of them is matched with the other which is the stigma's sporophytes, then the incompatibility happening. Clear? So in that means this is the rough diagram. You can see here, this is the pollen and this produces the two sporophytes containing the S1 and S2. Clear? And this is the carpel, and here the S allele that produce two kinds of sporophytes. One is the S2 and S3. So S1 is when transferred to the carpel, the pollen tube is not formed. Why? Because here the sporophytes are S1 and S2, and here the sporophyte is S2 and S3. So what I was saying, that one of two S allele matches, if matches, that means here the S1 is not produced here. You can see that S1 is not produced here, but S2 is matched with that. Clear? So S1 easily can identify that S2 is already produced here. So that's why the incompatibility is not happening. In the second case, you can see that the S1 and S2, from this S1, S2, the S2 is transferred here. That S2 formed by this carpel, this S2 sporophyte is transferred to the stigma, and where the sporophytes produced S2 and S3. So S2 is matched with the S2. So here also the incompatibility occurs, or pollen tube formation is not seen. And in the third case, you can see here that here S1, S2, so S2 is transferred here, and the two sporophytes that are produced, the S3 and S4. So S3 and S4 are totally different from S2 and S1. So that's why the sporophyte from the pollen that easily can identify that here the sporophytes that are produced S3 and S4 not be matched with the S2, and so incompatibility is not occurring here, and as a result pollen tube formation is happening. Clear? So this is the thing, that sporophytic incompatibility. Clear? In SSI, that male determinant is what? The male determinant is expressed in the diploid cells of the anther, tapetum, and their products added to the pollen as a constituent of the pollen coating. So what is saying that in SSI, male determinant that is expressed in the diploid cell. Where it is expressed? In the diploid cell of the anther. Anther means, which one is the anther? You all know that anther and filament are the two parts of the male reproductive system of the flower. So this is the anther. The anthers, so here the male determinant that is expressed by which one? The diploid cells present on the anther, and it's produce, this diploid cell that produces one product that is added with what? Added with the pollen as a coating, pollen coating. Clear? And this is the example in case of brassica. The male determinant is the cysteine rich protein located in pollen coat and is called the S locus cysteine rich protein. So what is saying that the male determinant is a cysteine. This male determinant is one kind of protein, and this protein is known as the cysteine. Okay, this is cysteine, and this is located where? This is located in the pollen coat. Clear? That means this pollen coat is totally formed by this cysteine protein. Clear? And this is called the S locus cysteine rich protein. It's a short name is that SCR, and full name is that S locus cysteine rich protein. And the female determinant is which one? This is the serine or threonine receptor kinases, and this is called the S locus receptor kinase. So kinase is another kind of protein. Clear? And this is located where? In the plasma membrane of the stigma cells. So I repeat this one again, or try to explain that the two proteins here important. One protein that is present in the pollen as the pollen coat, and second protein that is present in the stigma of the carpel. And the first protein that is the which one? The S locus cysteine rich protein. Okay, and the second protein that is known as the S locus receptor kinase. The two proteins here important, the short form is that SCR and SRK. So which is located in the plasma membrane of the stigma cells, and this is located where? This is located in the pollen coat as the that is in the pollen, you can say in the pollen, and binds only to the cognate SCR, and SRK, this binds with what? This binds with the SCR. The two proteins, they bind with each other, and if this happens, that means the binding is possible, then SCR to SRK, that causes the autophosphorylation. So when binding of SCR and SRK that possible, then autophosphorylation occurs, and this phosphorylation of the SRK receptor that initiates signaling cascade, and that inhibits the pollen hydration and germination. So what is saying that if the phosphorylation of the SRK receptor that initiates the signaling cascade, and that inhibits the pollen hydration and germination. So here the important thing is that the binding of SCR and SRK, because if they bind with each other, then a signal form and phosphorylation occur, and due to this signal formation that inhibits the pollen tube formation or germination and hydration obviously, because pollen tube needs water that is absorbed from the stigma for increasing its length or for extending its length. Clear? So this is the thing that is happening here. And here the three general ways is seen in this type of incompatibility. The first one is that the germination of the pollen may be decreased. It may be possible. So here this is seen in the broccoli. And the second, where the pollen germination, that is normal and that is occurred in normal way, but the pollen tube growth, that is inhibited in the stem. Totally this is inhibited. Pollen tube growth is inhibited. In third case, the incompatibility reaction occurs after fertilization. So what are the three ways? The three ways are, first one is the germination of the pollen may be decreased. Second is the pollen tube germination is normal, but this is inhibited in the stem. And the third way, where the incompatibility reaction occurs after the fertilization. These are the three ways by which the incompatibility process is occurring. Clear? And the examples are, the first process that is seen in broccoli, the second it is seen in the tobacco, and the third one that is seen is the Gesteria. So this is the Sporophytic Incompatibility. And I have to say one thing, if you are the newcomers, so please you subscribe if this channel is helpful to you, okay, and give a like for the video, and if this is liked by you, and you can also dislike it. And another thing is that you can comment in my comment section.